My name is Crystal Boggs. Hi, my name is Crystal Bly, and I am doing my transcription and translation video. I found this exercise really educational, trying to figure out the best way to understand, watched many videos, read many descriptions, and this is kind of what I came up with that worked best for me to made me understand kind of what transcription and translation was. Transcription is the process of RNA synthesis controlled by the interactions of promoters and enhancers. Several different types of RNA are produced, including messenger RNA, which specifies the sequence of amino acids in the protein product, transfer RNA, and ribosomal RNA, which plays a role in the translation process. It starts with transcription, which involves the 4 steps, which are initiation, elongation, termination, and processing into the translation process, which I like to think that there are 4 steps in that one as well. Again, initiation, elongation, termination, and producing actual protein that our body is constantly doing every day. So for transcription, the 4 steps, one is the DNA molecule unwinds and separates, forming this open complex where the RNA polymerase binds to the promoter of the template strand, leading to elongation, where the polymerase moves along the template strand, synthesizing the RNA molecule. It's a little bit different for eukaryotes and prokaryotes. So in prokaryotes, the RNA polymerase is a hollow enzyme consisting a number of subunits, including sigma factors that recognizes which promoter is being, I guess, recognized. In eukaryotes, there are 3 RNA polymerase, 1, 2, and 3, and the process includes this proofreading mechanism. Then termination occurs. In prokaryotes, there are 2 ways that that transcription is terminated. A lot of T's there. So row-dependent and row-independent termination. With row-dependent termination, there's this protein factor, rho, and is responsible for disrupting the complex involving the termination strand. Or I'm sorry, the strand itself, RNA polymerase and the RNA molecule. Let me take a drink. My dogs are going crazy, I apologize. In row-dependent termination, a loop forms at the end of the RNA molecule, causing it to detach itself. Termination in eukaryotes is a little bit more complicated, involving an additional adenine nucleotide at the 3 of the RNA transcript, and I believe it's oh my goodness, I forget the name of what that process is called, but it starts with poly. After that is the processing. So after the transcription of the RNA molecule, it's processed a few different ways. Introns are removed from the exons. The introns are removed and the exons are splitting, or not splitting, they're coming together. Oh gosh, what is that called? Splicing together to form a mature mRNA molecule. consisting of a single protein-coded sequence. RNA synthesis involves the normal base pairing rules, but the base thymine is replaced with the base uracil. And then translation comes into effect. Those 4 steps again: initiation, elongation, termination, and then the process, the very end, the product being the protein. Initiation is a small subunit of the ribosome binding at the 5. Oh my goodness. At the 5 end of the mRNA molecule, and it moves to the 3, where it meets the start codon and then forms the complex with a large amount of ribosomal complex units, with the initiation of the tRNA molecule. Then elongation takes place. In elongation, the subsequent coatings of the mRNA molecule determine which tRNA molecule is linked to the amino acids that bind to the mRNA. The enzyme peptide transferase, sounds so fancy, links to the amino acids to form this peptide bond, and then the process continues, producing a chain of amino acids, and the ribosomes move along this mRNA molecule to termination. Translation is terminated when the ribosomal complexes reach one or more of the stop codons. The ribosomal complex in eukaryotes is larger and more complicated than in prokaryotes. The process of transcription itself occurs within the nucleus, and the process of translation occurs in the cytoplasm, which provides more opportunities for gene expression, which I believe that we are getting to, or I will be getting to here very soon. I found this so interesting, like I was saying before, because this is happening in my body constantly, and I am amazed at what I am doing without even realizing it. So it's pretty neat, and I appreciate this exercise, so thank you.